think so. So I was going to say, yeah, I'm just going to get going. So thank you for inviting me, and thanks to Alan for bringing me up here, and especially thank you for removing all the snow off the ground before I got here. I'm a Southern California boy, as you can tell from going to Santa Cruz, so the snow thing is not really up my alley. Once this gets going, what I'm going to talk to you about today is a research project that I've started at UNC in close collaboration with Dr. Shahzad Sheikh, who's a PhD, MD PhD with a clinical focus in the inflammatory bowel diseases. It's been really a lot of fun and has been done sort of building on the infrastructure that at UNC within their Center for there we go. All right, so I'm on the first slide. There we go. So this is done within the context of the Center for Gastrointestinal Biology and Disease over at UNC that really has become a leader in the treatment of gastrointestinal diseases, but especially in inflammatory bowel diseases. Involved is a NIDDK, has a center grant. But importantly for the stuff that we're doing has been a great resource in being able to obtain patient samples. Shahzad has played a large role in developing a tissue bank and working with the surgeons there to give us the precious material to be able to run the experiments that we're trying to run and understand IBD from a more molecular perspective. All right, so. Well, if I knew which button to push. None of them seem to be. We were. Fortunately, I need to show you a little bit more than just this. So basic outline of my talk today, I want to start off and just go over a little bit about Crohn's disease so that you're familiar with the different aspects of it that'll be important for today's talk. I'm going to take a couple of slides and just describe this idea of cellular identity that I think is going to be very familiar to most of you, but this sort of set the stage for the two then different studies that will follow. One where we've looked at this in the context of a mouse model, the IL-10 knockout, and a second one where we were looking at patient samples, colon tissue from patient samples. And it's in that, as my title said, that we've uncovered these two molecular subtypes that we're very excited about. Okay, good. All right. So IBD, what is IBD? It's a big problem right now, and it's one of these diseases that in this Western, westernized part of the world seems to be becoming worse. Whether the incidence is increasing because we're getting better at identifying it and diagnosing it, or whether it's because a lot more people are contracting the disease, probably a combination of both, but it's becoming a large problem. And one of those that I read a statistic the other day that 20% of us have a relative that has it, and most of us probably don't even know it. This is a disease of young people. Most of the age of onset is usually in your adolescent years, your early 20s. And often these patients are coming in and they've been misdiagnosed over and over again. And by the time that they get to a facility like UNC, the disease has progressed. And these patients are just miserable. Okay. I'm just going to have to, I don't like standing behind the podium, but I think I'm going to have to. So the inflammatory bowel disease is actually an umbrella term. It's a collection of diseases. The two that we're most familiar with are Crohn's disease and ulcerative colitis. And I'm going to be talking mostly about Crohn's disease. But just to show that these are a heterogeneous set of diseases, ulcerative colitis is generally a general inflammation of the colon in a very contiguous way and envelops pretty much the whole colon. Whereas Crohn's disease is different in that it can actually affect tissue, as they say, from gum to bum, from your mouth to your anus. And it's primarily seen in the terminal part of the small intestine, the ileum, and then within the colon. And what's unique about this compared to ulcerative colitis is that what you will see is you will see patches of affected tissue that are right next to other tissue that look perfectly normal. And you will have this sort of patchwork presentation of the disease throughout the intestines. From a clinical standpoint, it's also a very heterogeneous disease. Both in the location, as I described, although primarily they classify this as being affecting the ileum, affecting the colon, or affecting both of them. But even when you look at the affected regions, there's multiple presentations from just a general inflammation to the formation of these ulcers or these little holes within the lining of the intestine, to a stricturing where you see this narrowing of the intestine that can cause blockages, to fistulas in which you actually, the intestinal wall is completely perforated and it can form these tunnels back to the intestine or to other organs of the body, including the skin, that obviously is not a good thing. Like so many diseases, this is a multifactorial disease. There are a lot of things that seem to be contributing to the onset and progression of this. Sort of when you ask someone to describe CD, they say this is an aberrant immune response to the intestinal microbiota in a genetically susceptible individual. So those three components are generally accepted as all contributing to that. What we don't know is how much each of those contributes and exactly why people get this disease. Generally, it's associated with some kind of environmental trigger, which is very not well understood. We know that the diet, smoking, other sort of environmental factors can increase the severity of the disease in ways that really are not understood. From a genetic standpoint, they have done GWAS on this, and I'm sure that a lot of you have seen these papers about the GWAS of IBD. This has actually been one of the very successful ones. We've got over 200 loci that have been associated with the disease, but similar to other GWAS experiments, almost none of these have been well characterized. The vast majority of them are in non-coding regions, and we really don't understand how they're contributing to the disease. More importantly, from Shahzad's standpoint and being involved in sort of getting the chance to be involved on the clinical side, all these GWAS experiments, the identification of all these associated loci have not really translated into the clinic. So when Shahzad sees a patient, his knowledge of where all these associated loci does nothing in how he treats his patient or his ability to improve the condition of his patient. So like so many other people, you know, this is what we're trying to do, is figure out how can we take all this knowledge that we're gaining and turn it into information that can help in a clinical setting. And so, and in addition to that, currently on the clinical side, there is a clinical classification system that's based on the location and on these different behavioral phenotypes. But even that system has not translated into knowing how to treat patients better or being able to predict the progression of the disease. So just because you present with general inflammation, that really doesn't tell you whether you might go on to develop strictures or fistulas, or whether you're going to respond to certain treatments or not. So, like in so many diseases, we feel that if we can understand Crohn's disease better at the molecular level, we might be able to predict or understand different clinical aspects of the disease, such as response to treatment, how it's going to progress, and whether there's a recurrence. So the focus of our studies has been doing this, is to really try to understand the disease from a clinical level. So I told you I wanted to spend a couple minutes talking a little bit about this idea of cellular differentiation and identity. So we all know that our genome's exactly the same in all of our cells. But each of our cells, the different cell types, have very unique functions and responses to external stimuli. And largely that's due to the expression of different genes and the expression levels of different genes that sort of dictate what these cells do. And taking a step back even from that, the expression level of these genes are then governed by the gene regulatory system, which is largely the epigenetic system that influences these gene expression patterns. So the way that we think about, one way to think about differentiation is at each step of differentiation, it's a reprogramming of this epigenetic state to change sort of the gene expression patterns to allow these cells to adopt new functionality. And so as you go down to the different tissue levels, each of the cells in each of these tissues then have a unique epigenetic program that sort of dictates what that cell does. And so one way to think about disease is that now we have a condition where we have cells that are not behaving normally. Their gene expression programs have been altered in a bad way, and a lot of this may be then driven by changes in the epigenetic programs dictating this expression program. So thinking about this combination of epigenetics and regulation, I think we're all very familiar with the background of sort of genomics. On the epigenetic side, we can talk about this in terms of different types of epigenetic modifications, including DNA methylation, histone tail modification, and nucleosome repositioning and creating these areas of open chromatin where then transcription factors are allowed to bind. And these sort of define the epigenetic program that then influences downstream the expression of genes and microRNAs, which microRNAs can then come back and provide another level of regulation at a post-transcriptional level. And in this genomics era, we've developed assays that are able to measure these different molecular characteristics, both from an epigenetic and from a transcriptional point of view. So in my lab, and as Alan sort of says, a large focus on the epigenetic side has been open chromatin, which initially we started off with DNA-seq and FAIR-seq, and have now progressed to ATAC-seq. And so in the assays or in the experiments that we're going to talk about today, it's primarily going to be the combination of looking at open chromatin and the effect on transcriptional output through mRNAs and microRNAs. And for those of you that may not be familiar with open chromatin, though I'm guessing that if you've seen any of Alan's talks, you probably are. You can think of this simply as one of these high-throughput sequencing functional experiments where the output is an enrichment of sequences that indicate where regions of nucleosome-depleted open chromatin are. They're primarily found, or a lot of the strongest and the largest are found in promoter regions, as you might imagine, where all the transcriptional machinery associated with POL2 needs to bind. But also includes the more distal regions where these more distant enhancers and repressor elements are binding, influencing then gene regulation. So the main question that we wanted to answer initially is, do we find evidence that in Crohn's disease, is there a reprogramming of the epigenetic landscape that is contributing to the onset and progression of this disease? To first establish a potential role for chromatin in this, we started with a mouse model of colitis, which is the IL-10 knockout mouse. IL-10 itself is a cytokine that is one of the key anti-inflammatory cytokines by regulating NF-kappa B. And so it suppresses inflammation. So by knocking it out, these mice will spontaneously develop colitis in the presence of bacteria or in presence of a normal microbiota. In this particular study, we chose to look at one particular immune cell type, the macrophage, that is in the gut. And the reason why we're looking at macrophages is they play a key role in this inflammatory response as part of the immune system. The macrophages, though, within the intestine are very unique compared to macrophages that are found in the rest of the body, in that, like macrophages in the rest of the body, one of its main jobs is to identify and eliminate harmful bacteria. But if we think about a normal intestine, there are a lot of bacteria and other microbiota that live in the intestine and that are good bacteria, working in a symbiotic way to help in food digestion and absorption. So the macrophages in the gut do not want to mount the inflammatory response in the presence of this bacteria, because this inflammatory response would serve to damage the tissue. And so there needs to be a tolerance of this bacteria within the gut. And so the presence of this inflammation seems to indicate an aberrant activity of these macrophages in response to this bacteria. So to look at this, we looked at both wild type and IL-10 knockout mice, and we looked at peripheral macrophages, so bone marrow derived macrophages, to see what did a macrophage look like that is normally programmed to mount an inflammatory response, in addition to the intestinal macrophages that should be programmed not to mount this inflammatory response. All right, so there's animation that I don't think I'm going to be able to show you. So, because clicking on it's not going to work. Anyway, so our overall design is to look at open chromatin and transcription in these, the peripheral and intestinal macrophages from these two mice, and look at how they react in the presence and in the absence of bacteria. And sort of trying to look at this a little bit more concretely. So here I've shown some open chromatin signals from peripheral macrophages in wild-type animals, both in the presence and the absence of bacteria. And so what we're looking for are these changes based on the introduction of bacteria, or in looking at peripheral and intestinal macrophages, differences between the two of them. When we look specifically at the peripheral macrophages, both in the wild type and the IL-10 knockout mice, and in the presence and absence of bacteria, we found three distinct types of chromatin regions. The first two that were related to a response to bacteria. So these are chromatin regions that in both the IL-10 knockout and in the wild type mice either became more open on exposure to bacteria and the introduction of bacteria, regions that became more closed on introduction of bacteria that are seemingly regulating then genes that are important to the response to bacteria. But then we saw this interesting third class, which were regions that were more open or more closed in IL-10 knockout mice in general, regardless of whether bacteria was introduced or not. And if we notice here, the majority, looking at the peripheral macrophages, the majority are of this type, of the inducible type, that are opening up or based on the presence of bacteria. So then our next question was, do we see something similar in the intestinal macrophages, and how does that compare to these peripheral macrophages? So in the same way, we found these three classes again, those that became more open on the presence of bacteria. So these, though, are slightly different experiments, because now Intestinal macrophages are normally exposed to bacteria. So in this case, we had mice that were either raised conventionally, meaning in the presence of bacteria, or mice that were raised in a gnotobiotic facility where conditions where there was germ-free condition. And so as I was saying, so we had basically the same three classes where either more open in the presence of bacteria, closed down in the presence of bacteria, and then these IL-10 regulated class of element. But what I want you to notice is that in this case, the vast majority of these are in this IL-10 regulated class. So more chromatin changes were occurring based on the elimination of the IL-10, the absence of IL-10, than in response to the bacteria. When we looked more closely at these IL-10 regulated regions in the intestinal macrophages and compared these to the peripheral macrophages, what we found was that there was a large overlap between those that changed on the absence of IL-10 compared to those that were induced by the presence of bacteria in the peripheral macrophages, which suggests to us that the elimination of IL-10 is reprogramming these intestinal macrophages to now look more like what these peripheral macrophages look like, and thus taking on the characteristics of inducing this inflammatory response in the presence of bacteria. So then that raised the question of, well, so if we simply added back IL-10, could we reverse that phenotype within the colitis model, within the IL-10 knockout model? And actually, IL-10 therapy is something that has been tried in treatment of Crohn's disease. What we found was that while by introducing IL-10, we could change the expression of many IL-10 regulated genes and either upregulate or downregulate as they normally are when IL-10 is present. When we looked at the chromatin, though, the chromatin didn't change. So simply adding back IL-10 is not able to reprogram chromatin back to its original state where it can tolerate the bacteria. And so if you remove IL-10, it's simply going to revert back to its inflammatory state, which we think might be a reason why in these trials of IL-10 therapy, for the large part, they have not worked well. And we think it's because it's not able to reprogram these cells back to their normal state. It's just sort of a Band-Aid that temporarily can fix, but is not curing the underlying molecular situation. So summarizing this first part, so what we think we've shown is that the absence of IL-10 is reprogramming the chromatin landscape within intestinal macrophages. And one thing I didn't point out, but this reprogramming is happening even in the absence of bacteria. So even these mice that are being raised in the germ-free environment, we still see these changes in the chromatin environment. So it's not that IL-10 knockout mice are being exposed to bacteria and this is somehow reprogramming these cells. They're reprogramming from the beginning. During the differentiation process, something is going wrong, but for whatever reason, IL-10 that isn't involved in any kind of epigenetic programming, it doesn't modify chromatin at all. Somehow the absence of that is creating this incorrect programmed cell. And the consequence of this reprogramming is now these macrophages have lost their tolerance to the bacteria and induced this inflammatory response that simply adding back IL-10 cannot restore. So that's great to sort of establish this in mice, in a model, but what about human Crohn's disease? So using patients that, material from patients at UNC that Shalad's colleagues were able to obtain, we looked at colon tissue, and we looked at colon tissue that was from unaffected regions of the colon. So these are not inflamed by eye. Surgeons look at it, it looked like perfectly normal tissue, but from patients with Crohn's disease. And we also looked at another small cohort of patients that did not have IBD. I want to call them healthy, but they didn't have intestinal inflammation. And with these tissues, we looked at their mRNA, microRNA, and chromatin profiles. And normally when you perform expression experiments like this, you see that, all right, you often will do something simple like a clustering, a hierarchical clustering, or a PCA analysis just to see whether you see different patients that are clustering in different ways. And so we did this assuming that we get the Crohn's disease patients that would cluster together, and we would get the non-IBD patients that clustered together. But surprisingly, when we did this, we did find two very distinct clusters, but not clusters that initially made sense to us. We saw one group of CD patients that were clustered all off by themselves, like we expected. But another cluster that were more similar to the normal patients. And when we looked at the genes that were driving this clustering, we found something really interesting. We found that in this off group, away from the normal IBD patients, a lot of the genes that were being upregulated were genes that were normally only turned on in the ileum. Whereas when we compared the two groups between this off group and the group of CD patients that was near the normal, the non-IBD cohort, a lot of the genes that were upregulated there were genes that were normally only seen regulated within the colon. And this led us to sort of call these things a colon-like group and an ileum-like group. Now, initially, we were very skeptical, and we spent about 2 months going back and staring at slides and making sure, did we mess up on the tissue? Did we actually get ileum tissue? But after a couple of months, we convinced ourselves it wasn't a tissue mix-up. This was actually present. When we looked at the open chromatin landscape, we saw very distinct differences between these two groups as well. And open chromatin regions that were being opened and closed that were directly related to these genes that were being differentially expressed. We further looked using roadmap epigenomics data at histone modifications that were typically only seen in either ileum or the colon, and found as well that those genes, those chromatin regions that were associated with the colon-like group had histone modifications that were more specific to the colon. So your activating marks, your H3, K27 acetyl marks in those that were more associated, that were only present within the colon. But in our ileum-like subgroup, when we looked at, we saw histone modifications that were enriched that were normally only seen within the ileum tissue. So from multiple levels, we're seeing evidence that for some reason we've got this aberrant subgroup that seems to be acting very similar to a more ileal-like phenotype. And so we labeled these, like I said, an ileal-like group and this colon-like group. And so our next question was, well, these are adult patients. They were, the tissue that we used were from resections. And so these were patients where disease had progressed to a pretty severe state. They had been through a lot of different treatments that could have affected this cellular reprogramming. So what we wanted to know, is this something inherent to the disease, or is it just reflective of the state of these patients? So fortunate for us, there had been a study that had been done on a cohort of pediatric patients that were treatment naive. So the initially diagnosed had not gone through all these exposures. The disease had not progressed as in the adults. And we wanted to see whether we saw a similar pattern. And what we found was when we did the principal component analysis using data from those, I want to add into this that the pediatric patients, these samples were from the ileum now, whereas the adult patients, these were samples from the colon. When we did a PCA analysis on all the samples put together, we found that, you know, the initial separation was between the two cohorts, but on PC2, which divides in the adult population our IL-like and our CL-like group, we also saw a separation among the pediatric patients. It wasn't as distinct as in the adult patients, but we definitely saw patients that seemed to have a more colon-like phenotype, even though they were from the ileum, and others that had a more ileal-like phenotype. Where on the right as well, we see the black dots that are normal ileum. And we see that those are all towards the bottom of PC2, or on the negative side of PC2, sort of grouping again with our ileal-like subtype in the adult, and what we're suggesting is a more ileal-like subtype within the pediatric patients. And obviously, since we did this on using gene expression data from both of these cohorts, these were similar genes that were driving this phenotype in both sets of patients. So our next question is, well, so what are these genes doing? So we did a pathway analysis. I'm going to get a hang of this sooner or later. I know. And so anyone that's done a pathway analysis knows you come back with a lot of different interesting things to look at, but I want to try to distill this down a little bit for you. So on the left, we have our adult cohort, and on the right, we have our pediatric cohort. And what I want you to take away is there's sort of two different classes of pathways that we see different between the two. Immune-related pathways and metabolic-related pathways. And on the immune side, we see the different signaling pathways, including interleukin, interferon, the NOD2, toll-like receptor pathways, suggesting that the immune response between these two different classes of patients is slightly different. Now, when we did this analysis comparing both of these classes to normal or non-IBD patients, we saw, you know, there were definitely differences there that indicated that reflected the inflammatory response that was present in CD patients. But we're still seeing the differences between these two populations in their immune system response. The second are all these metabolic pathways, which isn't that surprising since this is such a metabolically active tissue. Both of them are metabolically active tissues. But what we see is we see differences in lipid metabolism that may be reflecting differences in the epithelial barrier that is made up of lipids and its ability to keep the enteric microbiota in the lumen and out of the yes, where it's not supposed to be. We also, I'm sorry, part of this is, you know, messing with my head. Anyway, so we also see xenobiotic metabolism, which is dealing with the foreign agents, the microbial presence within the gut. We see energy metabolism, and in both cases, we see upregulation in the TCA cycle. So we see a shift in energy metabolism towards the TCA cycle and a bit away from the glycolytic cycle. And so we see fundamental differences in activity within these two classes of patients. We also wanted to look at additional molecular characteristics of these patients. And so with the help of Praveen Sethupathi in his lab that is focused on microRNAs, we also look at microRNAs. And performing PCA analysis based on microRNA expression, we could exactly recapitulate our two classes, suggesting that this epigenetic reprogramming is also affecting the transcription of microRNAs. We dug deeper into the microRNAs that might be driving this response. Okay, so now we get everything all together. And we found, in particular, there was one microRNA, miR31, that seems to be much more highly expressed in our ileum-like cohort compared to our colon-like cohort. miR31 is normally very highly expressed in the ileum and is not expressed in the colon, which this reflects. Now we know that colon tissue is made up a lot of different which is reflected. So we actually see that miR31 is upregulated in even the colon-like phenotype. So the presence of disease seems to upregulate miR31, but it's really in this subtype of the ileum-like class, or the ileum-like patient, where we see this extreme upregulation in miR31. All right, this will be interesting to see what just happened. Okay, so just to make sure that everyone understands what do microRNAs do. This is like I said, a post-transcriptional regulator of mRNA levels. And these microRNAs that are transcribed target mRNAs by binding to generally target regions within their 3' UTRs that suppress, either target them for degradation or interfere with their translation into proteins. All right, I don't know. Okay. And the point of something you missed is that each of these microRNAs can target several different genes. There's not a one-to-one correspondence between these. And so what we want to know is, do we find evidence that miR31 is actually a master regulator of mRNA expression between these two subclasses? And the way that we looked at this is using a program that was developed in the Sethipathi lab called miRHub. And what we specifically wanted to look for were microRNAs that were upregulated, in this case, in our ileum-like subclass, whose targets were at the same time shown to be downregulated in that class, since we know that these microarrays are targeting and resulting in the downregulation of these genes. And so we looked at all microRNAs with all differentially regulated mRNAs. And of all of the microRNAs, it was, again, miR31 that stood out as the only master regulator, suggesting that its upregulation was actually contributing to the differential expression at the mRNA level between these two cell types. Okay. So colon tissue is a complex tissue consisting of several different types of cells, different immune cell populations, and epithelial layer. And so looking at this in colon tissue, and this is a problem with anyone that's looking at heterogeneous tissue, you really don't know what is the specific cell type in which differential regulation or differential programming is having the largest effect. And so in Shahzad's lab, they've been able to sort colonic tissue into multiple different populations, including macrophages, T cells, B cells, and the epithelial layer. In each one of those populations, we looked at cells derived from patients with Crohn's disease and patients without Crohn's disease, and we measured the miR31 level. And we found overall a general upregulation in miR31 in each one of the different cell populations. But as is very apparent on this slide, although it's not going to pop up, we have 100-fold upregulation in the epithelial cells, which in the microRNA world is almost unheard of. Usually you're looking for sort of a twofold induction of expression. But to see something induced to this level is pretty amazing. But so going back to sort of our original focus of this, it's all great to be able to do these molecular studies and find these out, but GWAS has not been able to contribute in a meaningful way to in a clinical setting, and so we wanted to know. So is knowing that these two subtypes exist, does that tell us anything useful about clinical characteristics of these patients? So we did an association with many different types of clinical characteristics with some that turned out to be significant, which I had highlighted, that you're not going to see. But essentially what we found was that those patients in the colon-like subgroups were those that were more apt to require a complete colectomy, that would have perianal involvement or rectal involvement, whereas those in the ileum-like cohort were those that were more likely to have ileal involvement. So this is, obviously a very small collection of patients. We only had 21 Crohn's disease patients and 11 non-IBD patients. And so we were amazed that we were even able to find these significant of associations even with this small number. Okay, so just summarizing the second part, what we think we've shown very convincingly is that there exist at least two molecular subtypes within Crohn's disease that have very distinct molecular profiles that are shown at the chromatin level, at the mRNA level, and at the microRNA level. And I haven't shown you, but it's also, when we looked at long non-coding RNAs, you also saw this. And all of these together are suggesting that the cells within these two different cohorts are being reprogrammed in very different ways, and especially in the ileal-like cohort in the adults, these are being very fundamentally reprogrammed to take on functions that are not seen normally in the colon. And very encouragingly, we did find that they were associated with different clinical phenotypes. So just to kind of wrap up a bit, so what I hope I've convinced you, and what we're trying to convince people, is that if you think about this from the idea of you have a cellular identity that is defined by your epigenetic program that leads to a particular gene expression profile, that in disease, often what you are going to see is you're going to see a reprogramming of this epigenetic landscape that is going to be changing the function of these cells and making them act in ways that they aren't supposed to. And in Crohn's disease, and particularly in macrophages, it's reprogramming them to contribute to this inflammatory response, this aberrant inflammatory response in the presence of microbiota, leading to inflammation and tissue damage within the cells. We know that there is a genetic component, and obviously we haven't done enough patients to be able to fully investigate the genetic component. We also know that the genetics can influence this epigenetic programming. So this, in our minds, can provide a potential link where we see, based on a genetic background, we're seeing a slight reprogramming of the epigenetic landscape that in the presence of the microbiota is now triggering this aberrant inflammatory response. So we're excited about this, and we've got a lot of big plans, which I hope to show you. First thing we want to do is we want to scale this up to a lot more people. We've been funded now to replicate these chromatin and expression experiments in many more samples to see whether we, again, can replicate these two subtypes that we initially find, but then also see whether we can find additional subtypes that might indicate slightly different molecular programming leading to different clinical outcomes. In addition to looking at tissues, we also want to look more at individual cell types so we can understand more from a mechanistic standpoint how these differences in the epigenetic landscape are contributing specifically to individual cellular reprogramming and changing their function, leading to this inflammatory phenotype. The enteric microbiota is obviously contributing to this, and so we have embarked on some microbial sequencing to see if we see differences in the communities that are present in not only CD versus non-IBD, but also within our molecular subtypes. As I showed, a lot of the pathways that are being affected in these molecular subtypes are related to metabolic processes. So we're gathering samples to send off to do some metabolomics to see if we see very distinct metabolic profiles reflecting this change in metabolic activity, both between just even CD and non-IBD, but between our molecular phenotypes. We've started working with IBD doctors at UNC that specifically treat pediatric patients, because a lot of people in the field feel like to really get at the root of the disease, we need to be examining patients that at initial diagnosis, before they've gone through these rounds of treatment and before the disease has been allowed to progress to these more extreme phenotypes, and to see whether we start seeing hints of this molecular reprogramming early on in these pediatric patients. And of course, you know, we want to perform more functional experiments to understand exactly what's going on in these cells. So in Shahzad's lab, they've been able to create colloids and enteroids, which is essentially extracting epithelial cells from these patients and being able to grow them up and form sort of these mini guts, mini organs that then we can experiment with using different microbial products and changing the expression of genes within these to see how they react, to give us a better sense for how this epigenetic programming may be affecting normal cellular function. So obviously, with a project like this, with many different facets, we have a lot of people that are involved. So in my lab, both on the computational and analytic side, and Shahzad's lab that are doing a lot of the wet lab experimentation, and the subtypes lab, including someone who's sitting in our audience that left Praveen's lab and has joined a lab here. And everyone's contributions are equally important in trying to understand these molecular underpinnings of this disease. And I think that's it. So if they have any questions.
My first question is about, in the first part, it's interesting to notice that germ-free animals also have this wrong identity macrophage. We cannot blame it on the wrong type of bacteria trigger over aggressive immunity. Is that generally true for human patients as well?
Well, as far as I know, we don't have any human patients that have been raised in a motobiotic facility. So it's a great question, and I don't know the answer to that. So from sort of our point of view, throughout the stages of differentiation, it's going to be affected by a particular environment that those cells are differentiating in. So for these cells that are entering the gut, what we feel is that the environment within the gut has changed so that now the epigenetic reprogramming is being changed based on that environment. Whether it's partly influenced by the presence of different colonies of bacteria, we don't know. But so if somehow we could manipulate, maybe one way to look at this is if somehow we could manipulate the bacterial colonies and see whether that somehow rescues a normal epigenetic phenotype. But to eliminate the bacteria. We do know that, yeah, you know, can't do it like you can in mice.
My second question is about how to tie up the parts. Because the first one is the mouse, it's about macrophage, it's about chromatin. The second part is human, it's about colonic cells and fibroblasts, and RNA and microRNA. So do you see, for example, the ileum-like subtype is related to the wrong macrophage in humans?
So it is hard to sort of relate the two, and really the reasoning behind the two studies is one, first in the mice is just a way to more definitively show that there is this reprogramming happening in the presence of a chronic intestinal inflammation phenotype. But we're not trying to say that the changes that we see there are necessarily going to be the same changes that are driving Crohn's disease in humans. We probably will see certain, you know, we do see some similarity in genes that are being affected. But it was really, it's sort of established in the field, and this is in the field of IBD, that we need to be looking at chromatin. We need to be looking at epigenetics. This is important in the disease. So that when we presented them with the human data, we can point to it and say, you know, sort of we have this precedent. But it does make you question how good are these mouse models of disease, and really how much can we learn in these mouse models that will translate into human clinical.
Sort of two related questions. Now that you have your samples subdivided into two groups or three groups, if you take the non-IBD people out, if you now overlay the 203 GWAS hits, is there anything that the 203 GWAS hits post factum are different? I mean, are there some that are sort of specific to some subtypes and not others? And related, if you look at the epidemiology or the clinical history, can you say that, you know, the kids that are in the ileum group are the ones that got five times ear infection and got bombarded with antibiotics, or anything in the history that might have something to do with the microbiome or with the environment that was changed?
Okay, so to answer your first question, when we look at, so it's been, like I said, it's been hard to study the genetics because we have such few samples. When we look at the GWAS hits, we look just overall at regions that are changed between CD and non-IBD. We've seen enrichment and loci associated with the GWAS hits in these differences between those two large classes. We don't really see an enrichment one way or the other when comparing, say, the ileolite subtype and the colon-like subtype. So we've been sort of reluctant to dive into the genetic field until we've been able to do this in many more patients. You know, in our small level, we just, you know, when we look at specific genotypes in these patients, we're not seeing any obvious correlations with GWAS loci. As far as relationships to sort of the history of the patients, this is why we think the pediatric cohort is so important. And really, the patients that are involved here are ones that we are getting samples before they've started any kind of treatment. They are just initially diagnosed. They haven't been put on steroids. So whether there was a history of antibiotics or in early childhood related to infections from other sources, they're not our patients, and we don't have access to that history. But there was nothing, at least at the time of diagnosis, that you know, these sets of patients were on antibiotics at that point because that's the way they were treating. So as far as we could tell, at least in the pediatric patients, there was nothing different about their history. But then, like I said, we don't have complete access to those. On the adult side, we did look through treatment histories, and we haven't found a connection to antibiotic use or steroid use or anti-TNF alpha type anti-inflammatory use.
So those are really exciting data. In part 2 of the talk, it looks like the reprogramming is specific to the colonic epithelium, correct?
So
At least with respect to MIR31.
with respect to MIR31, yes.
So,
yeah.
oh, sorry. So my question is, do you see the inappropriate expression of intestinal cell types in those ileum-like colonic samples? For example, Paneth cells that are like the stem cell support cells in the small intestine that don't exist in the colon?
So we get this question a lot of times. In this ileal-like subtype, is this just that, there's been this invasion of Paneth cells? We're very, there's this Paneth cell metaplasia phenotype that is often seen in these CD patients. When we showed these histologically, when we showed these to pathologists and they looked at these histologically, they couldn't see any difference. So we don't think that it's somehow this, just complete change by this invasion of these different cell types. So that answered part of your question, but I don't feel like I answered the first part of your question.
I wasn't even curious necessarily about the invasion of Paneth cells, but is there inappropriate trajectories of the LGR5 positive?
Right. So we haven't been able to look at the individual cell types. And so we really don't have an idea whether, the changes we're seeing are specific to a single cell type. If they're, the one that I did show, MIR31, obviously, we see a much larger effect in the epithelial cells. And when we look at the whole colon tissue, the vast majority of cells in the colon tissue are epithelial cells. And so it will be interesting to us to see, as we start being able to do these experiments in sorted cells, what changes do we see specifically to different cellular populations? What do we see sort of ubiquitously across those? And what might those be, suggesting about changes in specific cell type functionality that is contributing to this?
In the pediatric Crohn's disease patients, did you also see upregulation of 31?
We don't have microRNA data for those.
Got it.
So we actually have a project right now where we are generating microRNA data in a different set of pediatric patients to see whether we see, and we are seeing differential expression across these on MIR31. We haven't had a chance to really look at this in depth. It's not as clean cut as we see in adult patients, which we're not surprised given what we saw in the mRNA results. But that does seem to be differentially regulated across the pediatric population as well.
Cool. So have you guys looked at maybe some sort of motif enrichment that could lead? So I'm thinking of these differentially open chromatin regions, or even the GWAS hits. Do they seem like they're pointing to some developmental factor or some motif that at least you could do a ChIP-seq later on and narrow down on that?
Right. So, like good computational biologists, we did motif enrichment, and yes, you see motifs that are enriched. One of the ones that was interesting to us was AP1, but overall, nothing that is sort of like your dream. Hey, here's a motif that appears in all these regions, and it completely explains how they're being reprogrammed. It's just not that simple. All right. Well, thank you.